Well, do you know, we already made it to Wednesday here in Living Environment fans. And how was that algebra test yesterday? Did you take it? How did you do on it? Good. Regents, big deal around here in New York State. Anyway, our Regents is coming up in just a few days now, right? Like, it's on the 14th. Today's the 5th. So that's 9 calendar days until the Regents exam, including today. That's 7 class days. It's really come up quickly. So let's look at some more old stuff again and try to refresh our brains as we get ready for the big test.
All right.
Hey now.
Mr. Kovacs class. It's Mr. Kovacs class. You might learn a ton. It's Mr. Kovacs class.
It's interesting too.
It's Mr. Kovacs class.
And he's super cool.
It's Mr. Kovacs class.
Hey, it's Mr. Potato Head. And of course, potatoes are starchy things. We know they're made out of carbs. Yeah, let's review like those macromolecules. Remember, we learned all about the macromolecules, carbohydrates being one of them, the starchy things. Those are complex carbohydrates. And the sweet things are simple carbohydrates. And if you remember, we called carbohydrates, they were polymers. And when you break them down, you get monomers. In the case of polysaccharides, in case of carbohydrates, you get what are called monosaccharides. Most common ones is glucose, right? Yeah, you remember that. Right, and then we could put 2 monosaccharides together, you get a disaccharide. That's like sucrose, table sugar, right? You put the bonds together. You do that by what's called dehydration synthesis. You take away water, basically, 2 H's and an O from the both, and you wind up connecting to them by their O, and you have dehydrated the monomers into a dimer, or the monosaccharides into a disaccharide. And of course, we get polysaccharides, or the big, right? In biology, carbohydrates come in basically one of 2 polysaccharides. They either come as starches, like starch, right? Or they come in fibers, like cellulose. Starch is digestible by enzymes in human bodies, but fiber is not. So for the Regents, you gotta remember the simple carbs or the sugars, glucose more likely than not, right? It's gonna be the sugar you're gonna need to know. And then complex carbs are either fibrous or starchy, right? Starches you can digest. Use amylase, which is the enzyme found in your saliva of your mouth to digest complex carbohydrates, break them into simple sugars. We learned about the NAs, the nucleic acids, right? We got DNA and RNA, right? And we learned that the monomer of a nucleic acid is called a nucleotide, and it's basically made up of a sugar called deoxyribose, or ribose, depending on what monomer you're looking at, right? And then it's got a phosphate group, and it's got this nitrogenous base attached to it, right? And then it's the bases that make all the difference in the DNA code, right? Like, there's basically 4 in DNA, right? There's four nucleotides. There's A, T, C, and G. And remember, A goes with T, apples in the tree, cars in the garage. G goes with C. And hopefully you remember that. And of course, the important thing, of course, we learned was that these nucleotides make up the DNA code, and we will go over it a little later again, but like, remember, transcription is when you take the code and turn it into mRNA, and translation is when you take the code and, you take the translated code and you turn it into an amino acid sequence or a protein. Which leads us to our next lipids. Oh, you thought I was gonna say proteins. Yeah, we'll get to proteins, but lipids are one of the macromolecules, right? They're made up of monomers of fatty acids, which come in long chains of carbons. So really, they're an extension of carbohydrates, in that if you can take a lot of carbons and stick them together, you can store energy in the bonds of between carbons in a really efficient manner, and we call those fats, or sometimes waxes or oils, right? I mean, They come in different versions, but the lipids. Lipids are high energy storage molecules. They also, remember, phospholipids make up the cell membrane, and they are hydrophobic. They don't mix with water, so it's hard. They help to control what goes in and out of the cell, right? Steroids, like cholesterol is a good example, basically they can slip in and out of cells pretty easily because they are lipid soluble, right? And steroids are hormones usually, and cholesterol is not really a hormone, but it lives inside of cell membranes and helps to keep them nice and wiggly, right? And then like food lipids are usually the triglycerides. That's with 3 fatty acids stuck to a glycerol head. And so there's lots of lipids. Our last, maybe the most important to biology, are the proteins. Proteins are used for so many things. Their monomers are called amino acids, right? And proteins can be structural, they can be they can be used in hormones, they can be used in neurotransmitter, they can be used as receptor molecules, they can be contractile, they can be enzymatic, right? Like they can do enzyme functions and those kinds of things. Lots of different types of proteins. We learned lots of different types of proteins. We learned what was most important about a protein is its shape, right? And its shape is dependent on its amino acid sequence. And the amino acid sequence is put together during translation, right? Transcription is in the nucleus. Translation happens at the ribosome. Transcription and translation, right? And if proteins aren't the right shape, they can't do their job. And that always shows up on the reagents. They have to be the right shape. Proteins have to be the right shape. Good examples of proteins you should definitely know hemoglobin. You should probably know collagen and keratin. You should probably know insulin, right? Structural proteins usually end in IN, and then you should know some enzymes. Enzymes usually end in ACE, ASE. Okay? But what's important about them is their shape. You should also know that if they change shape, if their shape gets changed, it's called being denatured. And we learned that you could denature proteins by changing its temperature. High temperatures will denature proteins, and making the solution more acidic. Acidity can denature proteins as well. All right, and then of course, we'll spend more time later, but probably the biggest, most important group of proteins, I mean, you've got cell membrane proteins are pretty important, but the enzymes, right, the biological catalysts. We'll talk more about those at a later date. All right, so that was a quick review of all of those biomolecules. Remember, you get one crack at it today in your ed puzzle, and then we're going to do some practice regions questions. Fun. It's Wednesday.